We spend hours arguing about whether ancient humans ate more grasses or more meat, whether they used spear throwers or just their bare hands. But almost nobody stops to ask one of the most visible mysteries staring back at us every morning in the mirror. Why did we lose our fur? Look at your forearm. Now imagine that same skin covered in dense, dark hair like a chimp's. The difference is striking. Compared with most mammals and even our closest ape relatives, humans are unusually hairless across the body. At the same time, our skin is packed with sweat glands that pour out watery sweat capable of cooling us through evaporation. That pairing, remarkably bare skin and prolific sweating, is the biological fact any good explanation must account for. If you like deep science dives like this one, take a moment to like and subscribe. We'll be digging through several competing ideas, weighing the evidence, and staying honest about what we don't yet know. So, what selection pressures could have favored less body hair? There are several plausible pathways scientists have proposed, and they aren't mutually exclusive. Each offers a mechanism, some supporting evidence, and important caveats. Let's unpack the main candidates one by one, and then try to stitch them together into a coherent picture. One of the most widely discussed explanations is thermoregulation, often paired with the endurance running idea. Hair is a good insulator. On a cool night or for a furred animal that relies on radiative cooling, fur helps retain heat. But fur also traps a layer of air and moisture, and when you're trying to cool a hot body in a hot environment, trapped hair and insulating underlayers can work against you. Evaporative cooling, sweat turning into vapor, is one of the most efficient ways to lose heat. But for evaporation to cool effectively, sweat needs to evaporate from exposed skin. Heavy fur makes that process inefficient. So, one argument goes, in a hot, open environment where daytime activity could lead to overheating, having less body hair while developing lots of eccrine sweat glands for evaporative cooling provided a big advantage. The endurance running piece of this story brings behavior into the mechanism. Humans are unusually good long-distance runners and walkers compared with other primates. Features such as long legs, spring-like tendons in the feet, a nuchal ligament that stabilizes the head during running, and our ability to dissipate heat while moving point to selection for sustained activity in daylight. Persistence hunting is a classic example. A hairless body that can pour sweat on a large surface area would allow an early human to run or walk long distances during the heat of the day without overheating, giving an edge in tracking and exhausting prey or simply traveling across open landscapes. Comparative physiology supports part of this. Relative to many mammals, humans have a very high density of eccrine sweat glands that produce watery sweat well suited for evaporation, and our ability to lose heat by sweating is exceptional. But there are caveats. We don't get direct fossils of hair or sweat glands, so timing is fuzzy. Did hair thin before or after other endurance adaptations? Did these traits evolve on the savanna or in wooded mosaics? And even if thermoregulation played a major role, it's unlikely to be the whole story. Cooling is important, but hairlessness introduces other problems. Exposed skin is more vulnerable to sun and to parasites, so there must be offsetting benefits to outweigh those costs. Another compelling hypothesis focuses on parasites. Dense fur is an ideal habitat for ectoparasites. Ticks, mites, lice, and fleas find shelter, breeding sites, and protection in thick pelage. For many mammals, fur protects them from sun and cold, but it also invites an invisible cost, a higher parasite burden. Removing or reducing fur makes it easier to detect ticks or lice, to comb them out, and to apply behavioral or technological countermeasures. In social primates, grooming is a powerful anti-parasite strategy. Grooming removes parasites directly and can spread antiparasitic oils or behaviors through a group. Humans, as social animals, have elaborate grooming rituals and later invented tools to augment personal grooming. The logic is straightforward. In a social species where grooming and cooperative care are possible, losing hair becomes less costly because the group can help spot and remove parasites. And if parasites impose a significant fitness cost, illness, blood loss, disease transmission, selection could favor individuals with sparser fur who are easier to keep parasite-free. Comparative evidence bolsters this idea. Primates that groom more tend to have fewer visible parasite problems, and behaviors like allogrooming in chimpanzees are strongly linked to social bonds and parasite control. There's also indirect genetic evidence. Studies of human lice lineages suggest that head and body lice diverged in ways tied to changes in clothing and hair distribution. For example, body lice appear to have evolved when humans started wearing clothing, implying that clothing and hair patterns affect parasite ecology. But the parasite hypothesis has its own limits. For hairlessness to be selected for on parasite grounds, social grooming or other interventions must already be part of the species' behavior. Hair loss alone without grooming would make parasite problems worse, not better. So the parasite story likely relies on an existing social toolkit. A related and sometimes overlapping explanation invokes sexual selection and social signaling. Skin is a canvas. Bare skin makes scars, color changes, and small health indicators more visible. If healthy, unblemished skin reliably signals underlying health and low parasite load, then mates might prefer partners with clearer skin. Over generations, that preference could shift the population toward less hair. Hairless skin makes the signal clearer. This idea doesn't require conscious preference for hairlessness itself. It only requires preferences for cues correlated with health. Sexual selection can be powerful and fast, and it's often invoked to explain traits that seem costly in other terms. Testing sexual selection hypotheses is tricky because mate preferences change culturally, and fossil records won't show mate choices. Comparative data from other species where visual displays matter supports the general plausibility. Whenever physical traits reliably signal health, sexual selection can amplify them. Still, sexual selection is rarely a solitary explanation. It tends to interact with ecological pressures like thermoregulation and parasite load. A more controversial family of ideas centers on aquatic or semi-aquatic phases in our past, the so-called aquatic ape proposals. Proponents suggest that spending time in water could favor reduced body hair, for hydrodynamic reasons, allow buoyant locomotion, or facilitate foraging for aquatic foods. Some enthusiasts point to human traits like subcutaneous fat distribution and a tendency for infants to hold their breath as suggestive. Most mainstream researchers treat these claims cautiously because the fossil and comparative anatomical evidence for a sustained aquatic stage is thin. Many specialist critiques argue that the suite of aquatic adaptations in genuinely aquatic mammals, blubber, flippers, extensive nasal valves, is absent in humans. Put simply, aquatic ideas are energetic and intriguing but remain speculative and poorly supported compared with thermoregulation or parasite-based models. Finally, and crucially, we must bring culture back into the story. Humans are not passive vessels of genetic change. We modify our environment and each other's selection pressures through behavior and technology, a process biologists call niche construction. Tools, clothing, fire, shelter, and hygiene practices all alter the fitness landscape. Imagine a population beginning to lose body hair. At first, exposed skin would be at greater risk of sunburn, cold, and parasites. But if those humans can invent clothing, control fire to stay warm, use tools to remove parasites, or develop communal washing practices to clean skin and hair, they can buffer the costs of hairlessness. In turn, those buffering behaviors reduce the stabilizing selection that might otherwise favor fur retention. Archaeological and historical examples show how powerful cultural adaptations are. Consider ancient bathing technologies. Romans famously used olive oil and a scraper called a strigil to remove oil, dirt, and sweat from the skin before entering a bath. In many societies, communal bathing and specialized tools for skin cleaning became social institutions that made the management of skin and hair a public, not just private matter. These cultural solutions didn't cause fur loss millions of years earlier, but they provide a vivid analogy for how behavior can compensate for and amplify biological change. Similarly, combs, tweezers, and later soaps and detergents are technologies that allow humans to manage hair and skin in ways no other primate does. Putting these trends together suggests a multi-causal model. Thermoregulatory demands in certain environments created a selective advantage for better evaporative cooling. Parasite pressures favored individuals who were easier to groom and check. Sexual selection magnified visual cues associated with bare skin, and cultural innovations, tools, clothing, communal hygiene, buffered costs and shifted the selection landscape further. Under this mosaic, hairlessness is not a single adaptive answer, but an emergent property of interacting forces. both biological and cultural. Before we accept that neat synthesis, it helps to dig deeper into the physiological details that make the puzzle so specific. Human skin is not merely hairless, it's functionally different. Two gland types matter for our argument: eccrine sweat glands and sebaceous glands. Eccrine glands pour out watery sweat over large areas of the skin. That sweat evaporates and cools. Humans have an unusually high density of eccrine glands compared with other primates, and those glands are distributed over most of the body surface. Sebaceous glands, on the other hand, produce sebum, an oily substance that helps lubricate and protect skin and hair. Sebum makes hair less brittle and contributes to the skin's barrier function. In the furry animal, sebum is mostly deposited along hair shafts and helps condition the coat. In the relatively hairless animal, sebum spreads across the skin surface and interacts differently with the skin microbiome, the community of bacteria and other microbes that live on our skin and influence odor, infection risk, and immune function. Less hair changes the microhabitat for microbes and parasites, which can have knock-on effects for disease resistance and social perception. Odor cues visible signs of infection. This interplay between glands, hair, microbes, and behavior hints at why culture, cleaning, oiling, scraping, becomes so important once hair is reduced. Cultures develop practices to manage sebum, remove grime, and regulate odors. Historically, olive oil scraping or clays and soaps have been used to modify skin conditions that elsewhere would be handled by a fur coat. Comparative primate evidence provides useful tests for parts of these hypotheses. Many non-human primates retain thick pelage and rely less on sweating for temperature control. When they do sweat, it's often around particular regions and not the whole body evaporative cooling system humans use. Grooming networks among primates are intense and time-consuming. Grooming burns valuable time that could otherwise be spent foraging or mating. If grooming is costly, then a reduction in hair could be favored only if the net fitness gain outweighed grooming time, unless cultural innovations reduce grooming costs by letting tools and group cooperation substitute for hands-on grooming. Lice research provides an elegant example of how behavior, culture, and hair interact over time. Humans host different lineages of lice. Head lice cling to hair, while body lice are adapted to clothing and living garments. Genetic divergence times between head and body lice can be used as proxies for when widespread clothing use began, because body lice require clothing to persist. Those molecular estimates suggest clothing use tens to hundreds of thousands of years ago, long after our hominin ancestors first began to look more hairless, according to some interpretations. This indicates a complex temporal relationship. Cultural innovations like clothing can emerge as responses to changing skins and climates, and then themselves become selective forces in new ways. We should also look at the fossil and archaeological hints for timing. Hair doesn't fossilize, of course, and sweat glands leave no marks on bone. So researchers look for indirect clues. Anatomical features related to running, heat dissipation, and thermoregulation, distribution of hominin fossils across different climates, and genetics that reveal when humans may have lost certain types of hair or pigment. Some scholars place key changes in the Homo lineage at times when early Homo species were moving into more open, hotter environments. Others argue for a gradual process with different selective pressures in different places and times. The bottom line is that the chronology is uncertain. Hairlessness could have arisen in stages, with different populations and environments contributing different pressures. Let's pause here and think about what would have to be true for each major hypothesis to carry weight. If thermoregulation and endurance running were central, we should see a consonant suite of anatomical changes for locomotion and cooling in the fossil record, and comparative physiology should show that hairlessness materially improves evaporative cooling. We do see anatomical evidence for endurance capabilities in Homo, and we do have clear evidence that sweating is an effective cooling strategy. If parasite avoidance were central, we would expect that social grooming, tool use for grooming, and cooperative care become important early on, behaviors that are plausible for hominins with strong social bonds. If sexual selection was key, we would need evidence that visual cues of skin condition affected mate choice over generations. This is harder to test but consistent with general principles of sexual selection. If culture reshaped things, then archaeological signs of clothing, shelters, and hygiene practices should track with changes in selection pressures, kina, which some forms of evidence do. Because the best explanation is likely plural, it's worth imagining a plausible evolutionary storyline that threads these elements together. Picture a hominin population living in a mosaic environment, patches of woodland alternating with open areas. Climate oscillations periodically expand open savanna-like habitats. Individuals who foraged or chased prey during the heat of the day encounter overheating risks. Mutations that reduce dense body hair and increase eccrine sweating provide a thermoregulatory benefit, especially during sustained exertion. Those same mutations may also make parasites easier to detect and remove, an advantage in social groups that groom and care for each other. Meanwhile, any visual cue of health, clearer skin, fewer parasites, becomes a private signal of mate quality and is favored by sexual selection. As this biological trajectory unfolds, cultural solutions like clothing, the controlled use of fire for warmth, and grooming tools begin to emerge and spread, reducing the penalty of living with exposed skin and allowing hairlessness to persist and intensify in different populations. Over deep time, this creates the human package we inherit: relatively hairless bodies, plentiful sweating capacity, complex grooming behaviors, and a rich set of cultural practices to manage skin and hygiene. That integrated view helps explain otherwise puzzling facts. For instance, if hairless skin is good for cooling but bad for cold nights, why aren't we covered in more insulating fat? In part, our cultural technologies, clothing, shelter, controlled fire, allow us to survive colder conditions without reverting to thick pelage. That cultural buffering is crucial. It means that once some hair reduction happened, cultural innovations could make hairlessness viable in more environments, and cultural practices like clothing can themselves become selective pressures, for instance, by changing parasite dynamics or mate preferences. It also reframes how we think about modern behaviors. Today's hygiene rituals, deodorants, shaving habits, and fashion choices are modern manifestations of very old problems. How to manage skin, odors, parasites, and social signaling in a bare-bodied primate. Historical examples make this vivid. The Romans didn't invent body hygiene out of nowhere. They developed practices like oiling the skin and scraping it with strigils, tools that removed dirt, sweat, and oil, as a social and hygienic ritual. In other places, bathing cultures developed communal baths and soaps centuries before modern chemistry. These practices are cultural responses to dealing with exposed skin in social settings. They're not the reason we lost fur in the first place, but they are examples of how cultural innovation and biology co-evolve. What about modern consequences for health and the microbiome? Our skin hosts a complex microbial ecosystem that interacts with sebum, sweat, and hair. Reduced hair alters those ecosystems and the way odors are produced. Underarm odors, for example, arise from bacterial metabolism of sweat compounds in hairless regions. Cultural practices like washing with soap, using deodorant, or shaving change those microbial interactions. That's interesting scientifically and culturally, but it doesn't mean one grooming habit is universally natural or better. The evolutionary story explains why our skin is structured the way it is. It doesn't prescribe a single hygiene regime. There are trade-offs. Some microbial exposures may help calibrate immune responses, while others can promote infection. Modern lifestyles alter those balances in ways we're still sorting out. It's important to stress where evidence is strongest and where it's thin. Comparative physiology and anatomy offer solid support for the role of sweating and heat management in human evolution. We are unusually built for evaporative cooling and sustained activity. The parasite hypothesis is also well anchored in basic ecology and primate behavior. Fur invites parasites, grooming reduces them. Sexual selection is plausible and consistent with evolutionary logic, but harder to demonstrate directly in deep time. The aquatic ideas, while imaginative, remain on the margins due to lack of convergent anatomical evidence. Cultural buffering is not an alternative to biological explanations. It's a necessary complement, explaining how early hair loss could persist and spread despite the new vulnerabilities exposed skin creates. So why did humans lose their fur? The honest, evidence-minded answer is because a suite of interacting pressures made hairlessness, together with enhanced sweating and new cultural behaviors, the most successful package for many populations of our ancestors. Heat dissipation for daytime activity and endurance likely played a major role. Parasite avoidance provided complementary pressure that favored visible, groomable skin, and sexual selection could have amplified shifts toward bare skin. Cultural inventions ranging from grooming tools to clothing and bathing then reshaped the selective landscape, buffering costs and reinforcing the new trajectory. That conclusion doesn't erase the uncertainty. Timing is fuzzy, and different hominin groups in different places may have followed different routes. Evolution rarely produces single, neat answers. It weaves multiple threads into whatever fabric happens to confer an edge in a given place and time. Our hairlessness is less an on-off switch flipped for one tidy reason and more a tapestry. Biological changes, social behavior, and cultural technology all stitched together over hundreds of thousands or millions of years. If there's a final takeaway, it's this. Our skin is a historical document. The absence of thick fur, the abundance of sweat glands, the oils our sebaceous glands produce, the microbiome that lives on us, and the rituals and tools cultures invent to manage all of the above. Together they tell the story of how biology and culture co-evolved. When you shave, put on deodorant, or strip down for a summertime run, you're participating in a long, complicated conversation between genes, parasites, climate, and collective behavior. Which of these hypotheses do you find most convincing? Thermoregulation, parasites, sexual selection, cultural buffering, or a mix? Drop a comment with your pick, and check the description for a short reading list if you want to follow up on the primary studies and reviews that explore these ideas in more detail.